Okay, so here we are talking about chapter 6, and this is about acellular pathogens, specifically viruses, viroids, and prions. So A means without, cellular means a cell. So these are basically a unique class of infectious agents that basically kind of blur the lines of definitions. So viruses aren't alive. They're not considered to be alive because they cannot reproduce by themselves. But they are, as we'll see later, very, very good at adapting. And I don't know, it's kind of scary because we try to be a step ahead of them, but they are very, very tricky. So we're going to be focusing on the viruses, viroids, and prions, and basically these are pathogens that lack a cellular structure, yet they do possess the ability to replicate with in susceptible hosts. So again, that means they cannot reproduce on their own. They have to hijack another organism in order to hijack their reproductive system in that cell. So we're going to be talking about the structure, classification, and replication strategies, how they interact with hosts, as well as mechanisms of the disease. So acellular pathogens are infectious agents. They're not made of cells. So they don't have those organelles that we were just talking about in the eukaryotic cells. They don't have the cytoplasm or true cell structure. And because they cannot reproduce on their own, again, they are not considered to be living. Instead, they hijack the host cells or the host machinery in order to replicate. And so we want to pay particular attention to the viral life cycle, how diverse they are, how they can keep changing every single time that they replicate, or at least very rapidly, how they can cause persistent and latent infections, and how the public health significance of emerging viral diseases such as Ebola and HIV and COVID are front and center in our lives. So we need to understand the acellular pathogens and how they're essential for appreciating the modern challenges in infectious disease control, epidemiology, and therapeutic development. So talking about those main types of acellular pathogens, again, viruses is the one that we're most likely familiar with, and this is very important clinically. COVID virus is one of the ones that we are all very familiar with. We also have HIV, influenza, and herpes simplex viruses. And so the structure of them is that they have genetic material, which can either be DNA or RNA. They have a protein coat around them, which is called a capsid. And they also sometimes have a lipid envelope around them as well. The key features are that they are obligate intracellular parasites. This means that obligate means they have to take over another cell and kind of just infiltrate them, right? So again, break it down to those keywords. They have no metabolism of their own. Again, when they hijack that cell is going to live for them, it's going to reproduce for them. And they use the host's ribosomes, enzymes, and ATP. So again, it's kind of like a bank robbery. They're like, hold up, I'm taking over now, and I'm using everything that you have as mine. Again, those are some of the diseases. And again, they're not alive outside of a host cell, and they can't reproduce on their own, which means they are technically not living. Two things in life scare me, cancer and viruses are both of them, and that is because they are both uncontrolled cell reproduction. Um, Prions, this is where it has a misfolded protein structure. And so there's no RNA or DNA, but anytime that we have misfolded proteins or misfolded anything, it's not going to function correctly. So remember, as you continue on through your educational and career, form is function. If it's not formed correctly because it's misfolded, then it's not going to function correctly. So again, the structure of a prion is a misfolded protein. The mechanism is going to be that it causes abnormal proteins in the brain to misfold, therefore not functioning correctly. And the effect would be that it causes normal proteins, or I'm sorry, the effect is that it would be progressive and that can cause fetal neurodegeneration. So degenerate means a breakdown of the nervous system. Okay, and again, this is going to be lethal. Diseases with these examples would be like Creutzfeldt-Jakob disease and mad cow disease. Um, and they're extremely resistant to heat, radiation, and disinfectants. So, not so fun fact, but prions actually were a big thing in the early 2000s. And so, this is like again, the mad cow disease. And so, it was one of those things that they were able to trace it down to patient zero and say that one was like a brain surgery that was performed on that person. And then the next surgery was like an eye surgery, and the next surgery was like a hip surgery or something. And of course, they had sterilized all of the instrumentation according to the current standards. But what they were found after they figured out that this was a prion disease is that it wasn't efficient enough to kill the prions. And so in the early 2000s, they had to change the protocol for surgical instrument sterilization to be higher and also higher heat and also some other things so it can kill things such as prions as well. Then we have viroids, and that would be where there is the structure of them would be a small circular single-stranded RNA, and it does not have a protein coat, and the hosts are in plants only, and the impact is going to be very much on crop diseases. And so this is not a human pathogen, but it is great for like a trick question. So pay attention to, again, the viroids are going to be in the plants only. So here's a little quick comparison table where you have your genetic material of viruses, our DNA and RNA. Prions do not have any, and viroids only have RNA. Your protein coat is present in the viruses, but not in prions or viroids. The viruses and prions can affect humans, but viroids are only plants. All of them need a living cell so that they can hijack it. And then there's some examples for you as well. So, unlike bacteria, fungi, protists, and helminths that we talked about before, acellular pathogens do not consist of cells, and so they cannot carry out metabolic processes independently. So, remember, A means without, cellular is a cell. So, it shows that this doesn't have its own cell structure. And so, despite them being very simplistically built, they are responsible for some of the most severe and globally significant diseases known to humanity. And so viruses in particular have shaped public health history throughout pandemics such as influenza, HIV, AIDS, COVID-19, which we're still in, and the Ebola virus disease. And so with chapter 6, we talk and introduce more of those acellular pathogens by exploring their discovery, what makes them different in their structure, and replication strategies, as well as interactions with host organisms, which highlights why they remain difficult to treat and control. So in figures 6.1 through 6.4, we talk about the general characteristics of viruses. And so viruses are infectious acellular particles that functionally obligate as intracellular parasites. Again, this means that they hijack a cell. So it's an intracellular parasite, it's a parasite, it takes over controls, and it's not a symbiotic relationship like we've talked about before. It is parasitic, so it's only benefiting that virus. They lack the ribosomes and the metabolic enzymes and the ability to generate energy independently. Therefore, they again have to hijack that cell to do everything for them. They do consist of a nucleic acid genome, either DNA or RNA, but they never have both. And again, it is enclosed with a protein capsid. Um, some viruses possess an additional lipid envelope that is made from cell membranes from the host, and it's embedded or it's made with these viral glycoproteins that's used for host cell attachment, and that comes into play when we're talking about like identification and replication. Because viruses rely entirely on the host cell machinery for replication, they are also going to exhibit strict host specificity, and tissue tropism. And so basically, the viral attachment depends on the presence of those specific host cell receptors. And so this explains why certain viruses can affect only a particular species or a particular cell type, and they don't necessarily affect all. And these features underscore why the viral infections can be very difficult to get rid of or eradicate without damaging the host tissues. Again, they take over and they hijack the cell, and then again, the receptors aren't the same. So again, it can cause some difficulties in targeting. When we're talking about viral structural and morphology, we're going to talk about some of the different shapes. And so this is talked about in figure 6.3 to 6. yeah, 6.3 to 6.6. And so we can have those helical viruses, such as like a tobacco mosaic virus, and this can possess those rod-shaped capsoids or capsids that are formed by repeating protein subunits. It is going to be built by having the capsoid protein wrap around the genome in a spiral, and it can form a rod or a filament-shaped virus. And some of those key points would be that the genome length determines the capsoid length. So if it has a long gene, then it will have a long capsoid length. In animal viruses, those helical capsoids are always enveloped or enclosed with another membrane, and these helical viruses are more common in RNA viruses. And some of the examples could be influenza, which is both helical and enveloped, also Ebola and rabies. If we have icosahedral viruses, these are going to have symmetrical polyhedral capsoids that effectively enclose the viral genome. And it's built with 20 triangular faces, and they look roughly spherical, and they're very stable, and they have a very efficient structure. And so the key points is that they can be naked or non-enveloped or enveloped, and the most common capsoid shape in human viruses, and they are going to protect against the genome in harsh environments. And some of the examples would be like adenoviruses, which are naked, poliovirus, which is naked, polio, and herpes virus, which is both enveloped and icosahedral. Okay, I paused for a second to put some pictures in. Sorry about that. All right, so then we have again those isochondral. They have 20 triangular faces. They look roughly spherical, and they are very stable and an efficient structure. And they can be naked or enveloped. They are the most common capsoid shape in human viruses, and it's going to protect the genome with the harsh environments. Some examples would be adenoviruses, which would be naked, poliovirus, which is naked, and herpes virus, which is both enveloped and icosahedral. And then we have our complex viruses, and these are represented by like bacteriophages. They display elaborate structures, including heads, tails, and tail fibers used to attach themselves and also then to be able to like inject that genome. And then they do not fit purely helical or isohedral icosahedral symmetry. They do have different structures or add-on structures in a way, so they can have a head and a tail and lateral bodies. And well, the structure is kind of complex. We have some pictures coming up. So key points is that they often have multiple components for attachment and delivery of that genetic material. And the structure is more function driven, not necessarily shape driven. And these do include some of the largest viruses, such as bacteriophages. And remember, to phage means to eat, and bacteria, it would be bacteriophages. So they infect and eat bacteria. The icosahedral head is where the genome is, where the helical tail with the fibers, that's going to be the one that's actually going to penetrate the cell wall there and inject the DNA. We also have pox viruses, and these can infect humans and animals, and these are brick-shaped. They are enveloped but not icosahedral, and they replicate in the cytoplasm, and this is a very high yield. And like I said, I have some pictures coming up here. And then the last one is the enveloped viruses, and these are included in like the influenza virus and HIV, and they acquired their lipid envelopes during the budding part from the host cell membrane, so they steal it. The viral glycoprotein spikes then embed in the envelope, and they are very, very necessary for the host cell recognition and entry. And so basically, it's the one that's going to like hold out the sign that says, "Here I am, this is what I am," and it lets them therefore be a target for vaccines and antiviral therapies. And so, what does enveloped mean? It just means that the virus has a lipid or a fat membrane that is taken from that host cell. And then that envelope is going to surround the capsid, which is like the outer capsule, right? And that can be either helical or icosahedral. And basically, they're going to envelop, the envelope contains viral glycoprotein spikes. So, again, this is something that can be used for identification and targeting. These ones, thankfully, are fragile and they can be destroyed by soap or like rubbing alcohol, heat, and drying. And but they can spread with close contacts from like fluid and respiratory droplets, like sneezing on somebody or spitting or something. So some examples would be HIV, influenza, herpes simplex virus, and SARS. So HIV, I mean, this one, don't get like scared that if you touch somebody there, you're going to get it, or if they sneeze on you. It's very low chance of it, but this is more like, close contact, they sneeze on you, you shake hands and stuff. This is where you can get like SARS and influenza, but like HIV and herpes is harder to get than just like touching a door handle. Back in the 80s when they first started researching it and stuff, they thought that it was that easy. And then, fun fact, Princess Diana, God bless her soul, she gave like a HIV patient like a big old hug on camera, and everyone was like mortified because they're like, oh my gosh, you're gonna get HIV. But she just believed in compassion. And, obviously we have proven throughout time that it takes a lot more to infect a person than just a hug. Okay. So then we have our comparison table where we can have the shape. So if we have our capsoid symmetry in helical, it's going to be spiral. On icosahedral, it's going to have 20 sides. Enveloped is going to be variable, and complex viruses have none or they are mixed. In the helical envelope, they often have them, usually in animals. Icosahedral sometimes have an envelope. Enveloped always has an envelope, hence it's in the name, and complex sometimes does. Structural add-ons, there are none in helical or icosahedral, but the enveloped is going to have those spikes. And then the complex can have the tails, fibers, and the lateral bodies. Our typical hosts for helical are going to be our plants and animals. Icosahedral, all of them. Enveloped would be animals typically. And then our complex is going to be bacteria or animals. And then an example of helical is going to be influenza. Icosahedral is going to be adenovirus. The enveloped is going to be HIV. And then complex would be an example of like bacteriophage or pox viruses. So some takeaways: complex viruses are not helical or icosahedral. Bacteriophages have a head and a tail. Pox viruses, they are complex and enveloped, and they also have a cytoplasmic replication. And envelope present does not mean that it has a capsoid shape. And so here are some pictures of what we can expect. And so this would be the helical. So you can see that there's like this helix, right? The adenovirus is going to be the icosahedral or the polyhedral. So you can see it has like just more sides to it. Enveloped it's covered in like an envelope or kind of a protective barrier. And then complex is there are a lot of them just kind of stacked on each other. And so it makes it more complex. Okay. And then with these guys, what's interesting is like they use these tail fibers and stuff to like adhere to the cell that they're trying to take over. And then it's kind of almost like a syringe. So after they adhere, then this goes and it like goes and it pushes this spike down into the actual cell. And that's how it then puts its DNA into the hijacked cell to take it over. Scary. Some classification and taxonomy of viruses. So this is going to be like your figures 6.7 through 6.9 and tables 6.1 to 6.2. And the viruses are classified based on the genome type, the capsid symmetry, and the presence of the envelope replication strategy, like we were just talking about. And so it's going to introduce this Baltimore classification system. And basically this groups viruses according to like what their nucleic acid and messenger RNA and how it's produced. And they include double-stranded DNA viruses, single-stranded DNA viruses, and double-stranded RNA viruses, both with positive and negative sense single-stranded RNA viruses and retroviruses. And we have some pictures and talking about those more coming up. And then this classification framework is going to give us insight into how the viruses replicate and also like different strategies and guides on how we can develop antiviral drugs. For example, retroviruses require reverse transcriptase to convert RNA back to DNA. And so this is a process that is absent in human cells, and therefore we can use it very much as a target. So we can only go from DNA to RNA, from transcription to translation. We can't go backwards. And so again, that reverse transcriptase would be going from RNA back to DNA. We as human cells can't do that. So again, great target. So here are the Baltimore classes, and then there's a picture of them right here in a summary table. And so group one is double-stranded DNA, and this means it's transcribed from DNA, and the host can either be it can from be either the host or from viral RNA polymerase. Examples would be like adenovirus and herpes virus. Yield. This means that it uses the host nucleus, and so they can very much create a lot of them. Group two is going to be single-stranded DNA. So you go from single-stranded DNA to double-stranded DNA to messenger RNA. Example would be parvodog viruses, which is unfortunately common in dogs. And they must first make double-stranded DNA before they can reproduce. Group three would be again a double-stranded RNA, and the mRNA is a viral RNA dependent RNA polymerase, and it is abbreviated RDRP. So it's RNA dependent RNA polymerase, RDRP, and an example of this would be rotavirus, and the host cannot read the double-stranded RNA. Group four is going to be a single-stranded RNA, and so it is a positive sense RNA, and basically the genome is already in the mRNA. And so an example of this would be poliovirus, SARS, and CoV-2. It is going to be immediately translated upon entry, so it immediately gets to work as soon as that virus takes over. Group four is going to be a single-stranded RNA, and this is a negative sense RNA. And so it must carry that RDRP into the host. Examples would be influenza, rabies, and Ebola. This is also high yield, and so it must bring the polymerase to the party. Group six would be positive single-stranded RNA with reverse transcriptase. And so basically the RNA turns into the DNA. Again, that's the reverse transcription. Usually transcription is DNA to RNA, so reverse would be RNA to DNA. Then it is from the integrated DNA. An example of this would be HIV, and retroviruses are also part of that. So group seven, this is double-stranded DNA with reverse transcriptase. So it is a DNA, but it uses RNA intermediate. So it just kind of uses it to go from step one to step two. And it is from the DNA after the mRNA comes from the DNA after the reverse transcription because we went backwards. And examples would be hepatitis B. Also, it is partially DNA and then part RNA life cycles. So it kind of bridges both of them. So here is a little one look summary table of what we just talked about and some little memory hooks. Positive is ready, negative needs help. So it's going to bring the friends to it. Retro goes backwards, and double-stranded RNA is unreadable, so it must carry that RDRP. Here's just kind of a picture of class one double-stranded, class two, class three, class four, etc. So just so you can kind of visualize a little bit. So when we're talking about the viral life cycle in prokaryotic hosts, we have the life cycle of our virus here, and it's talked about in figure 6.7 and 6 through 6.9. Um, and so this is going to be a series of different stages. So we have the attachment stage, then the penetration, and then we have the biosynthesis, and then we have the maturation, and then the release. So in bacteriophages, these steps are shown through this lytic cycle, and to lyse means to break apart. So this means viral replication ends in the host cell breaking apart and like the regular cell and the release of the progeny virons. So like the ones that they took over and like just the new little viruses. So again, remember they're hijacking the cell, they're making it make copies of the viral DNA, and then they're going to be releasing it. And this progeny is like the first replication of that. And also, like a little bit different, some bacteriophages can undergo a lysogenic cycle, which can integrate their genome into the host chromosomes as a prophage. So it can get pretty complex. So again, viruses scare me. Some of the example or the cycles, we have the attachment or the absorption, and this is where the virus binds to that specific receptor on a host cell surface. and this determines the host range and the tissue tropism, and it can be used to cause capsid proteins, which are naked viruses, and they can also have an envelope glycoprotein spikes, which we talked about spikes a little bit before, and that would be enveloped viruses. And so a little clue, if the receptor isn't present, then an infection cannot occur in an attachment virus. And so the penetration, this is where attachment stage, sorry. With the penetration, this is when that virus is going to enter that host cell, and it can be by membrane fusion. So again, this would be through an enveloped virus. The two pieces come together and then they kind of meld into one. And then endocytosis, as well as that direct injection that we talked about before, and that would be from like bacteriophages. And often this is followed by uncoating or kind of removing that protective layer so that then the virus can go in and completely take over. We have that biosynthesis, and this would be from replication and protein production. And so this is when the viral genome takes over and it causes the replication of just the virus. And then these viral proteins are made using the host machinery, again, because they hijacked it. And it depends, the strategy depends on what classification it is. So it's either going to be DNA versus RNA, positive versus negative, and possibly reverse transcription. So again, basically viruses do not make their own ATP or ribosomes. Again, they're going to hijack that the original cell. Then we have the maturation or the assembly, and this is where those newly made viral genomes are going to be packed into those capsids. And this is structural proteins usually put together themselves on the outside into the infectious virions. And this often occurs in the nucleus. So this is where many DNA viruses replicate and mature, or it can also happen in the cytoplasm, which is where most RNA viruses or pox viruses are. The release is going to be like that last part where that lysis, again, to lyse means to break apart. So that cell is going to burst. And this is common in naked viruses, where that budding can be where the virus exits through the host membrane. It is typical of enveloped viruses, and the cell may survive temporarily, but in the end, it is going to die. And so, just kind of another thing that you can remember is that envelopes can bud off. Here's a quick little clinical comparison. So again, the feature, naked virus versus enveloped virus. The entry in a naked is through endocytosis. Through enveloped is going to be infusion or endocytosis. The release is for naked is going to be lysis, where enveloped is going to be budding. That cell damage in naked virus is immediate, where it's often delayed in the enveloped virus, but again, in the end, it is going to die. The stability of a naked virus is very high, where an enveloped virus is very fragile, and things like washing your hands will kill it. Okay, so good. So lysogeny allows for viral genetic material to be passively replicated alongside the host genome, and then it remains dormant until a stress trigger causes it to enter into the lytic cycle, and then it's going to be like, oh, wake up, I'm going to really hijack now. So this mechanism contributes to genetic diversity through transduction, which is a process where bacterial genes are going to be transferred between cells by bacteriophages. Lysogeny, again, just a little bit more. This is where the viral DNA integrates into the host genome, and this is called a prophage. Then the host cell survives and divides normally. The viral genome is then passively replicated and with the host DNA, but again, it's kind of like you don't know that you have it because it's just kind of sitting dormant, and there's no immediate cell damage because again, it's kind of sleeping or a dormant infection. Then it can switch to that lytic cycle. And again, to lyse means to break apart. And so this would be like environmental stress triggers that can trigger activation, like UV radiation, nutrient deprivation, chemical damage, etc. So this is where the prophage excises from the host DNA. And then the virus is going to enter the lytic stage, which is going to cause for replication, assembly, and that cell break apart or that cell lysis. And the exam phrase that you can remember is stress-induced activation. So like a lot of times like herpes simplex virus, the person doesn't necessarily know that they have it, but then if they're under a very stressful situation, it can cause an outbreak. So like cold sores is one of the herpes simplex viruses, and so that would be one that would follow that. Why it matters is that it allows for that long-term viral persistence. Again, a lot of times the person that has it, they don't know that they have it. And it can also alter or change the bacterial phenotype. And so that's called lysogenic conversion. And phenotype, pheno means like photo. So genotype is the genetics of something, where phenotype is what it actually looks like. So my phenotype is brown curly hair and blue eyes, right? But my genetics are lots of letters. Um, and an example would be like a toxin production in some bacteria in some bacteria. And then this can increase genetic diversity among different bacteria as well. So here is just kind of a little thing of that lytic cycle. So again, the virus comes in and it hijacks and it puts its DNA in the cell. It causes it to replicate and put together new viruses, and then it bursts open that cell or it lyses it, where the lysogenic cycle again is going to be just like the replication, but the lytic one is where it's going to lyse it and break it apart. So lysogenic versus lytic, I know lyse is in the word, but lytic would be the one where it actually breaks apart the cell. Um, with transduction, we have different types of where it transfers bacterial DNA between bacteria by using bacteriophages. And so we have generalized transduction, and this occurs during the lytic cycle. And this is where random bacterial DNA is mistakenly packaged together, and then any gene can be transferred. We also have specialized transduction, and this occurs during lysogeny, and this is where only the genes that are next to the prophage insertion sites are going to be the ones that are transferred. So this one's more specific and it's less random, hence the word specialized. So ways to remember it is generalized is like, oh, just in general, we'll do it to any gene. Where specialized is like, oh, we're only going to like transfer our specific neighbors or adjacent cells. Our quick comparison here is going to be that we have our viral state in the lysogenic stage is going to be dormant or in the prophase, where the lytic cycle is going to be active replication of the virus. Host cell in the lysogenic is going to survive, where in the lytic it's going to be lysed or broken apart. Trigger is going to be for lysogenic stress, which can then turn it into the lytic. And then the lytic is going to be immediate infection. And then gene transfer is specialized for the lysogenic and generalized for the lytic cycle. So lysogeny, again, is going to allow for silent replication of viral DNA within a host genome, and it promotes genetic diversity through bacteriophage-mediated transduction. So again, here is just a visual summary of the generalized transduction versus specialized transduction. We'll make that a little bit bigger so that when I send these guys out, you can see it a little bit better. Okay. Viral replication in animal hosts. So this is going to be talked about in your book in figures 610 through 612. And animal viruses follow similar stages of infection, but they enter the host through mechanisms such as receptor-mediated endocytosis or membrane fusion. And basically, once they're inside the cell, that uncoating of that virus is going to release that actual viral genome, which then can direct the synthesis or the creation, making of that viral protein and the nucleic acids. And then the genome type determines the replication location, with most DNA viruses replicating in the nucleus and most RNA viruses replicating in the cytoplasm. Retroviruses represent a unique replication strategy. For instance, HIV is going to use reverse transcriptase. So remember, we're reversing transcriptase. So transcription is DNA to RNA. Reverse transcription would be RNA back to DNA. And then the resulting viral DNA is then going to, again, take over the host genome as a provirus, which is going to allow for long-term persistence and chronic infection. Little overview on how animal cells or animal viruses replicate. They have the same core stages as the viral life cycle, but the entry mechanisms and replication sites are going to be different based on their structure and genome type. So again, those stages are going to be the attachment and the entry or penetration, the uncoating, the biosynthesis, and the assembly, and then the release. Okay. Talking about entry into those animal cells, the animal viruses, again, cannot directly inject their genome into the cell, unlike the bacteriophages. Instead, they have to enter either through receptor-mediated endocytosis, membrane fusion, or undercoating. So receptor-mediated endocytosis is going to be where that virus binds to a specific host receptor. The host membrane engulfs the virus, and then also this is common for naked and enveloped viruses. Our membrane fusion, this is going to occur only with the enveloped viruses, and the viral envelope fuses with that host cell membrane, and then the capsid is able to enter directly into that cytoplasm. So again, if there's no envelope, there can be no fusion. The uncoating, this is where the viral capsid is going to be removed, and then that genome is going to be released into the cell. And this marks the beginning of the viral control over host machinery. And biosynthesis, this is where the replication happens. So this is going to be, the location depends on the genome type. If it is a DNA virus, then it's going to replicate primarily in the nucleus, where it's going to use that host DNA-dependent RNA polymerase. An exception to this would be the pox viruses, which would be the cytoplasmic replication, and that's what would be the difference. RNA viruses, this is going to replicate primarily in the cytoplasm, and this is going to require RNA-dependent RNA polymerase, and those host cells don't have enzymes to copy RNA from RNA. So basically, you have DNA that goes into the nucleus, and the RNA goes into the cytoplasm. When we have retroviruses, these guys are unique because, again, they don't take their regular rules to heart. So basically, the key steps are that the viral RNA genome enters the cell, and then it's going to make it go backwards. So the converts the RNA back to the DNA, which is not a normal process. And then that viral DNA takes over and it integrates into the host genome. And then so now that's a provirus. And so it basically replaces your genetic material saying, oh, this is your new DNA. Awesome. And then this host cell then transcribes, which is it's going to take the RNA into the protein. So transcription is DNA to RNA. RNA to protein is translation. And so it's going to take the viral genes during the normal transcription and it's going to change it. The clinical significance, this allows for long-term persistence where the viruses stay around forever and they can lead to chronic infection. And then that viral genome is passed to daughter cells during mitosis. And so basically this is now your new genes. And so you have your high yield comparison table here, which I like. You have your DNA viruses, your RNA viruses, and then your retroviruses. So DNA viruses replicate in the nucleus, RNA in the cytoplasm, and retroviruses are in the nucleus after reverse transcription. DNA often don't have specialized enzymes where RNA has the RD to RP guide that we've talked about, and the retroviruses have the reverse transcriptase. There is no genome integration in DNA viruses or in RNA viruses, but in retroviruses, yes, it does integrate itself into the actual genome of the person. And then the infection type is either acute or latent for the DNA viruses and acute for RNA viruses. And for retroviruses, it is going to be chronic. So animal viruses enter the host through endocytosis or fusion. Again, they're going to uncoat and release their genome, and they're going to replicate in those locations that are determined by their genome type, with those retroviruses uniquely integrating the DNA into the host genome for persistent infection. I know that some of this is a little bit repetitive, but again, these are all kind of complex words, at least to me, and ideas. So repetition sometimes helps it sink in. So you could have your replication level where you have your acute infection. Acute means short term, like right now. Chronic means long term. So you can have, your primary infection, and then you have the clearance of the virus and virus infected cells. This would be like the flu. You have latent infections, where this would be your primary infection, and then you can have a continued replication of viruses and virus-infected cells. And then you can also have a latent infection where it can lay dormant for a while. And so you can have that primary infection and then it lays dormant. You don't have any signs and symptoms. And then you get signs and symptoms again. Then it goes dormant, signs and symptoms, etc. And this is also going to include viral genome maintenance. So again, not all viral infections kill the host right away. Some of them remain in the body long term, and they use different strategies to evade immune clearance. And then these patterns explain lifelong infections and reactivation of disease. Okay, so we have some persistent infections, and these are the ones that are persistent. So they are continuous, low-level viral replication. They have minimal or mild syndromes and for long periods, and most host cells often survive. Some key features is that the virus is never fully cleared, and there's going to be ongoing immune stimulation, and therefore causing gradual tissue damage. Classic examples would be hepatitis, where it can have chronic liver inflammation over years. And so some of the keywords for this guy would be low-level replication without severe symptoms. We have our latent infections, and these are where the viruses goes into a dormant or an inactive state. And so there is no active replication between the episodes, and there is periodic reactivation that can cause symptoms. Key features are where the viral genome persists in the host cell, and it can be reactivated by stress, illness, or immunosuppression. Examples would be like herpes virus, either oral or genital lesions, and varicella zoster virus, which would be like your chickenpox turning into shingles. And again, your memory hook would be latent, is silent, and then it turns into a sudden flare. Some kind of chronic infections, basically what's happening is that you can have sustained viral replication and it's a progressive disease over time. And then this does increase host cell damage. And key features are that the virus actively replicates for years and then the immune system becomes exhausted or impaired. And so it just kind of breaks down over time. And so then the disease severity does worsen over time. A classic example would be like HIV. So this would be where it's the gradual destruction of immune cells, and then that can turn into full-blown AIDS, which is a progressive immune system damage. So I do want to kind of point out a little bit HIV/AIDS and basically how much the research has gone over the last couple of years, and it's absolutely amazing. So HIV/AIDS used to be a death sentence, and it is no longer, which is awesome. A few years ago, Charlie Sheen came out as HIV positive, and I think he's full blown AIDS too, and he's still living and thriving. So I love medicine, and I love how science is. All right, so here is a little bit of a comparison again: persistent versus latent versus chronic, and the different features. Um, Here is talking specifically about plant hosts, and this is not figure 615, but it is referenced in 615. And so basically you can have where you have the infection of a plant, and then you have the seeds that are infected, and then you could have an infected seedling, and then it is going to be infection of leaf and the tissues, and then it actually develops into it, and then it can kill the plant, etc. Interesting little side note. My sister-in-law is a botanist. She has her PhD in botany and she runs the herbarium at Central, Michigan University, and she also has a flower farm. So she grows tubers, so like the flowers that have like the bulb that you plant in the ground. And a few years ago, she had a virus in one of her tubers, and so she had to throw all of them out because as a plant breeder, she can't put those out and there can't be any idea or any threat of it at all. So it's interesting because you wouldn't really think of this as like a big thing. It's like, oh, well, viruses are bad in people, sure, but in plants, pfft. But then, there's like tobacco viruses and other like potato blight and stuff that can take out whole entire crops and it can be incredibly damaging. So, plant viruses are very similar in structure and replication, but they lack virus-specific receptors and membrane fusion mechanisms. So, as a result, plant viruses rely on physical breaches to enter the host. So, basically, right here is there a knife is cutting things into it, or like a crack, or something like that. So, how it enters, the primary would be mechanical damage, like we were just talking about, wind, farming equipment, cutting things, handling or grazing, animals graze, deer, rabbits, things like that. And then also from insects, so aphids, whiteflies, leafhoppers, those are all ones that can also transmit the disease. And plant viruses do require a wound or a vector, though, and these would be how those wounds come about. You can't put a band. Well, actually, you can put a band-aid on cells. I think that splicing like different rose bushes together is just, it's fascinating. I don't know. Genetics is really cool. Often you can have cell-to-cell spread. So once inside, the virus is spread through the plasmodesmata, and this would be the cytoplasmic channels that connects the plant cells to each other. And then this viral movement of the proteins are going to enlarge the plasmodesmata to allow the genome passage. And this enables a rapid local and systemic infection. And so plasmodesmata is basically a plant virus highway. Replication occurs inside the individual plant cells, and they use host ribosomes and enzymes. And most plant viruses have RNA genomes. Common disease manifestations, this would be from where they plant viral infections that can cause non-lethal but growth altering effects such as stunted growth, leaf discoloration such as mosaic patterns or yellowing for chlorosis, leaf curling or deformation, reduced fruit and seed production, and these effects definitely directly reduce crop yields and quantity. So I am from a farming family, and my family still does definitely farm. And yeah, we have to watch out for these diseases, and that can be catastrophic. Um, Agricultural and economic importance. There are major causes of global crop loss, and it is difficult to treat once the infection is established. And so, basically, how we try to control it is to manage the vectors or have resistant plant varieties or prevent mechanical transmission. And the plant viral diseases are managed, but they are not cured. So here is kind of a quick comparison of plant versus animals. So cell entry in the plant, ooh, wow, plant is either mechanical damage or vectors, where in the animal it's either a receptor, a fusion, or endocytosis. Cell-to-cell spread is through the plasmodesmata, and where in the animals it's going to be the cell release and reinfection. Typical outcome would be either growth defects for the plant viruses, where in animal viruses, it's going to be cell death and persistence. The treatment is going to be prevention only, where in animals, we can use antivirals limitedly. Limitedly, is that a word? It is now. Plant viruses enter through, again, the wounds or insect vectors, etc. Okay, moving forward, viroids and prions. So again, viroids and prions, these are acellular infectious agents. So A means without and cellular is a cell. So they basically challenge the classical definitions. So again, I don't know, viruses in general are terrifying to me. I've said that about 4 times now. But basically, they don't really fit in any category. They act as though they're living, but they can't reproduce on their own, so then they're technically not. But basically, the classic idea is that a pathogen has to contain DNA or RNA within a cell or virus, but these guys don't necessarily. So they kind of tell us and show us that a disease can arise without cells and even without a nucleic acid, which is bonkers to me. So those viroids, what they are small circular single-stranded RNA. They don't have a protein coat, they don't have a capsid, and they don't have an envelope, but they are in plants only. Again, this is viroids, not viruses. And how they can cause disease is that they're going to interfere with the host gene regulation. They disrupt the normal RNA processing and protein synthesis, and they can alter plant growth and development. Some of their effects on the plants are going to be stunted growth, leaf distortion or discoloration, and they can definitely give you a reduced crop yield. And so, viroids, here's another thing, is like naked RNA, and they give you plant disease. Prions, these are infectious proteins only. So there's no DNA and there's no RNA. And how they cause a disease is there's a normal host protein that's forced into the abnormally folded shape. So remember, form is function. If we aren't shaped correctly, we're not going to function correctly. So this causes us to misfold the proteins, which again changes the shape, and then that's going to cause progressive neuronal damage. Okay. Examples again are Creutzfeldt-Jakob disease and other transmissible spongiform encephalopathies. Key features are, again, this is kind of a little repeat. They're extremely resistant to heat, radiation, and disinfectants, and they are always fatal. And they do have a long incubation period. So again, here's your little kind of summary. They cause protein misfolding, causes malformation and malfunction, and it can cause the breakdown of your brain tissues or your neural tissues. So here is just a little quick little summary table. Again, your feature between both of them. So public health and ethical concerns. So it basically is going to talk about why we care. Okay. So emerging viral diseases such as Ebola and how virology can connect directly to public health, ethics, and global security. So the outbreaks aren't just biological events, they're also medical, social, and ethical challenges. So we have on over the left here, we have zoonotic transmission. So this is where can go from an animal to a human. So not all diseases are zoonotic. So for instance, fleas, we can't get fleas from animals. Okay, and so this would be an example of that. And then our human-to-human transmission, and then we can also have the outbreak where it can cause death and surveillance, etc. So again, this is just to show that it's not only in plants or animals. Sometimes it can spill over into humans, and then it can cause global problems. Okay. So our emerging viral diseases, so these are basically newly identified or rapidly spreading viruses. Again, often they are zoonotic, which means that they can be transferred from animals to humans. And these often have a high mortality and limited treatment options. So this is why we keep researching them, right? So Ebola is a very good example. It can cause severe hemorrhagic fever, and so hemorrhagic means bleeding, right? So you're going to kind of start bleeding out with a very high fever. It is a high case fatality rate, and it is spread through direct contact with bodily fluids, and it requires strict containment. And also emerging viruses test health systems before science can catch up. We saw that with our coronavirus, right? And COVID. We were on lockdown until we could try to catch up. And thankfully, we had been working on developing vaccination before because COVID has been around for years, but that's a whole nother discussion. All right, so importance of rapid detection. Again, early diagnosis is going to limit the spread. Again, this is why we went on lockdown in the pandemic, because we wanted to try to keep everything in one area and delay spread. The delays do allow for exponential transmission and healthcare system overload, which we did see with COVID. It also requires laboratory capacity. So getting our COVID tests took a while to do and get our results back. You had to have specially trained personnel and use a lot of PPE so that they could try to not infect themselves. And then also transparent reporting, where I think that there was some reports that other countries really weren't reporting the correct numbers to make it seem like they weren't as affected from COVID than they were. And then again, early detection is going to help us with outbreak control. So infection control measures. So of course, it's going to be critical for protecting our workers, our communities, and our global populations. And so we want to isolate and quarantine, use proper personal protection equipment, contact tracing to trace it down to patient zero. And then also, if you do have mortalities, then how to safely bury and handle that whole thing. So, for a while they were like not wanting to embalm COVID patients or, have funerals, et cetera, because they weren't exactly sure what would cause transmission. And so COVID was interesting times. I think we've blocked a lot of it out of our memory already. But basically, failure in infection control can lead to rapid amplification of outbreaks and lots of very quick spreads. Also, we have some ethical considerations where our outbreaks often require decisions before the definitive evidence exists. So, experimental treatments. So, again, the COVID shot, it was highly recommended. Not everyone got it. Totally your own choice. This is not a pro or con conversation. But, at some point we were a little bit guinea pigs, but we also weren't because there was many, many, many years of experiments happened before they actually released the vaccine. But again, when you did get the vaccination, you had informed consent under crisis conditions. You also had to have equity and access to care. So I know they prioritize elderly, infants, and sick, but everyone, no matter of socioeconomic status, et cetera, had to be given the same chance to get that vaccination. Also, you can have balancing the individual rights versus public safety. So, like some of the healthcare workers were forced to get the COVID vaccine because they considered it to be public safety and they overruled your individual rights, or else you got terminated. So that's a whole ethical thing too. So basically, ethics become urgent when time and data are limited, and sometimes decisions are made for us. So wrapping up, today we have our global impact and international cooperation. So basically, viral epidemics highlight that pathogens ignore borders. They don't care. They go where they want to go to. Local outbreaks can become global crises, and no single country can respond alone. Like for instance, they know that COVID started in Wuhan, China, right? And it wasn't just a Chinese issue; it was all of us. We had to all come in and work together to try to, reduce death and mortality and morbidity. Effective response depends on global surveillance networks, data sharing, ongoing research, and international coordination. Again, this means all data shared, so no hiding data. Our takeaway is going to be where our emerging viral diseases case studies underscore the necessity of rapid detection, strict infection control, and ethical decision making, as well as a sustained global cooperation to help mitigate or reduce the impact of viral epidemics. Um, and our acellular pathogens are pathogens occupy a unique space in microbiology. They exist between chemistry and biology. Unlike cellular life, viruses and viroids and prions lack independent metabolism and cellular structure, so they're not technically alive, but they do exert outsized influence on living systems through their interactions and host cell molecules. Across diverse structures and replication strategies, these viruses exploit host molecular machinery. They evade or reshape immune responses. They drive diseases in humans, plants, and animals, and they influence ecosystems, agricultural, and global health. So we need to kind of think about these things and understand how the genome type determines the replication strategy, why host pathogen interactions shape disease outcomes, how immune defenses, persistence, latency, and evolutions intersect between them, and why emerging and re-emerging viral diseases remain a global threat, i.e., they're constantly changing. It's not a one and done. It's oh, it's out. Now we have to monitor it because it's going to keep mutating. So altogether, these concepts reinforce the central role of molecular biology, immunology, and public health in making sure that we can work against infectious disease. And so, although acellular pathogens are structurally very simple, their biological and societal impact is great. Okay, so it makes understanding the concepts behind them and the different classifications and how to try to attack them very important for students and healthcare professionals. And then here is some of the structure again that we talked about. We have the head and we have the neck and we have the collar, the sheath, that tail fiber, and the base plate. So basically, this is kind of like the chunk where it just injects itself. So the tail fiber attaches itself to the surface, and then this whole thing just kind of injects itself, where it's going to just inject that DNA, kind of like a insulin pen, right? You click it and it goes and it goes and it takes over. All right, so next up are going to be those questions. I'm sending those out on a separate document so that you can work on them, and then we will, I'll send out an answer key later. So, all right, so thanks for joining me for chapter 6, and I hope that you guys have a fantastic day. Thanks.